The recent advancements in cognitive neuroscience and behavioral neurology have theorized that interoceptive signals are the core modulators of negative emotions. Interoception is the perception of sensations internally, such as feeling thirsty or the need to breathe, and is largely suspected to be convoluted with facial emotional recognition. Despite the suspicion of this circuitry, there is a lack of evidence of how interoception readies facial emotional recognition in healthy populations and is non-existent in populations with neurodegenerative diseases. The links that have been established have mainly been captured through partial associations between separate interoceptive and emotional tasks. Due to this, it causes a failure in the true revelation to whether interoception can directly influence emotional processing at various neurocognitive and behavioral levels, which limits theoretical and clinical developments. However, this can be achieved through priming paradigms that highlight the connections between interoception on other social cognition processes and cardiac dynamics on negative facial emotional recognition, which in turn can potentially reveal the direct links between interoception and emotion. To capture the interoceptive effects on facial emotional recognition, we captured neurophysiological, neuroanatomical, and functional connectivity measures with the task capturing interoceptive effects on facial emotional recognition in 4 groups, 1 healthy controls, in patients with neurodegenerative diseases such as behavioral variant frontotemporal dementia, Parkinson's disease, and Alzheimer's disease. We measured online EEG modulations of the heart evoked potential in associations with brain structural and resting state functional connectivity patterns to evaluate whether and how interoception primes emotions. Behavioral evidence from healthy controls suggests that interoception increases sensitivity to negative facial emotional recognition. Alterations in both domains are systematic in behavioral variant frontotemporal dementia and less noted in Parkinson's disease, which contrasts the interoceptive pattern in general emotional deficits seen in Alzheimer's disease. Evidence suggests that the insula and the anterior cingulate cortex, or the ACC, are the key regions involved with integrating interoception in the emotional processes. This is supported by the salience network that includes both the insula and the ACC. The salience network is involved with processing internal events, which includes interoceptive and affective signals. This causes the salience network to take on a cross-modal role, which differentiates it from other networks that deal with emotional and exteroceptive processes. The salience network is damaged in patients with BVFTD and compared to healthy controls in Alzheimer's disease patients. And the connectivity with striatal hubs correlates with disease severity in Parkinson's disease. A priming paradigm is utilized in many studies to evaluate how priming occurs in the brain. Participants are asked to evaluate or respond to a stimulus that is followed by a neutral, negative, or positive prime. For this study, the interoceptive priming task consisted of first comprising a priming phase that included counterbalance interoceptive and control exteroceptive conditions, and then a facial emotional recognition phase after each priming condition. This was designed in our study because only partial associations have been made between separate interoceptive and emotional tasks. They fail to reveal whether interoception can directly influence emotional processing at different behavioral and neurocognitive levels, but this can be achieved through a priming paradigm. We advanced 3 sets of hypotheses. First, we predicted that HCs would show enhanced negative FER in the post-intero condition and that such effect would be selectively disrupted in BVFTD and PD patients relative to the HCs, contrasting with generalized FER alterations in AD. For BVFTD, such disruptions would be accompanied by impaired interoceptive accuracy. Second, we hypothesized that the HEP modulations would significantly increase during post-intero negative FER in HCs, but not in BVFTD and PD. Third, across all groups, we expected that post-intero negative FER would correlate with the volume of the insula and the ACC, as well as the connectivity along networks including such hubs. Instead, post-extero negative FER was expected to correlate with EN connectivity. Moreover, we predicted that associations between post-intero negative FER and SN connectivity would be abolished in BVFTD, and that no other group would show this specific pattern across networks and conditions. We recruited 114 participants, which reached a power of 0.95. To determine this size, we ran a power estimation analysis on G-star power 3.1. Of the 114 participants, 48 were healthy controls, 19 with BVFTD, 25 with Parkinson's disease, and 22 with Alzheimer's disease. Each patient was sampled based on gender, age, and education, and matched with healthy controls. The diagnoses were established through extensive examination and revised by neurodegenerative disease experts. The priming task consisted of two phases, a tapping priming phase and a facial emotional recognition phase, where patients were asked to identify the emotion of static faces. The tapping priming phase consisted of 4 blocks of 2 minutes each, and it was counterbalanced across all participants. The order of presentation was also counterbalanced per subject per group, meaning half of them started with interoceptive and the other with exteroceptive. Immediately after each tapping priming block, a block of the FER was administered, which yielded 2 blocks each of the post conditions. But for the test, patients had a 2-minute practice phase for each part of the task. High-density EEG, or HD EEG, recordings were also obtained during the FER blocks for each patient. For the tapping priming phase, a validated heartbeat detection, or an HBD task was used. Participants were required to tap a computer keyboard along with their heartbeats, or along with an external simulated heartbeat for 2 minutes. Accuracy score, D', was estimated for each condition per subject based on heartbeat detection outcomes via signal detection theory. The FER phase was based on Ekman and Freisen's images. In each of the 4 blocks, 56 faces were presented in a pseudo-randomized order per block for 200 milliseconds each. This comprised of 8 neutral, 16 positive, and 32 negative faces. The participants were asked to judge as fast and as accurate as possible, and this is known as the IES that was calculated per condition and emotion. 100 participants received HD EEG signals from the FER. This was comprised of 18 BVFTD, 23 Parkinson's disease, 18 Alzheimer's disease, and each group had healthy controls to match it that equaled an N number of 41. MRI and fMRI acquisition and pre-processing steps are reported as recommended by the Organization for Human Brain Mapping. In each center, following the standard protocols, we obtained 3D volumetric and 10-minute-long resting state MRI sequences from all patients. Recordings were performed in 3 scanners. For the resting state protocol, participants were asked not to think about anything in particular, to keep their eyes closed, and to avoid moving or falling asleep. We chose the closed eyes modality to avoid noisy signals coming from the visual cortex. The final images were demographically matched. Compared to healthy controls, BVFTD patients showed selective impairments of interoception. No significant difference was observed in Alzheimer's disease or Parkinson's disease patients relative to the healthy controls. With exteroception, no significant difference appeared in any group of patients in respect to the healthy controls. Results confirmed a compromise of interoception in BVFTD patients only. To establish a link between interoception and emotions, we examined whether interoceptive priming improved FER outcomes, which is a normal IES, in healthy controls. Relative to post-extero condition, post-intero yielded a better IES only for negative emotions, with a t-value equal to negative 2.63, p.01, and a Cohen's d value of 0.27. No difference was found for the other 2 types of emotion. We also tested this effect in each emotion across patients compared with healthy controls. For negative emotions, significant differences between groups in post-interoceptive FER was found. Specifically, post-hoc comparisons demonstrated significantly lower emotional recognition in the post-interoceptive condition related to healthy controls that had a mean of negative 0.48 and a standard deviation of 0.67 in all of the patient samples. In contrast, while significant differences were found between groups in the post-exteroceptive negative FER, post-hoc comparisons revealed deficits in only the Alzheimer's disease sample that had a mean of 0.61, standard deviation of 0.71, and a p-value less than 0.001, relative to the healthy controls that had a mean of negative 0.33, standard deviation of 0.62. The other groups did not exhibit significant differences against healthy controls and post-extero outcomes. For positive emotions, no significant group differences were found in post-enteropositive condition, but only in Alzheimer's disease patients that had a mean of 0.92, standard deviation of 1, and a p of 0.02 exhibited lower outcomes than healthy controls that had a mean of 0.09 and a standard deviation of 1.15 in post-hoc comparisons for post-exteroceptive emotional FER. For neutral emotions, significant differences were found between groups in post-interoceptive neutral FER. Based on the time window detected in healthy controls, we compared mean HEP modulation for negative minus neutral phases across groups. In the post-interoceptive condition, the negative neutral HEP difference was higher for healthy controls than BVFTD that had a W value of 251, P of 0.02, and a Cohen Z value of 0.33. It was also higher in Parkinson's disease that had a W value of 348 and a P value of 0.03, and a Cohen's value of negative 0.48. But it was similar in Alzheimer's patients that had a W value of 413, a p-value of 0.18, and a Cohen's value of 0.26. The post-exteroceptive condition yielded no significant differences between healthy controls in any patient group. Analysis for all groups revealed significant associations between post-interoceptive negative FER and the volume of emotional interoceptive regions, which is the right insula and the left rostral ACC. No significant associations were found between the post-exteroceptive negative FER groups. For all groups, seed analysis revealed significant associations, which means the better the performance, the higher the FC, between post-interoceptive FER and SN, that had an R value of negative 3.9 and a PFDR value of 0.009, whereas post-exteroceptive FER was significantly associated to the EN with an R value of negative 0.57 and a PFDR value of 0.007. No associations were significant within the control networks, such as the DMN, VN, and MN. In the tandem between healthy controls and BVFTD patients, no network was associated with post-interoceptive performance. However, post-exteroceptive FER of negative emotions was significantly associated with FC in the EN with an R value of negative 0.32 and a PFDR value of 0.014. An opposite pattern was found in the tandem between healthy controls and Parkinson's disease, with significant associations between post-interoceptive negative FER and FC in the SN, with an R value of negative 0.45 and a PFDR value of 0.08. No significant associations between FC and post-exteroceptive performance were found. Finally, the healthy control and Alzheimer's disease tandem presented significant associations between FC and SN and post-interoceptive FER, with an R value of negative 0.43 and a PFDR value of 0.008. This was also shown between FC and EN and post-exteroceptive FER, with an R value of negative 0.41 and a PFDR value of 0.008. We investigated the multimodal impact of interoception on negative emotions and the manifestation it takes across neurodegenerative conditions and healthy controls, and behaviorally, interoceptive accuracy impairments were only observed in BVFTD. Interoception primed negative FER in healthy controls in Alzheimer's disease, despite the Alzheimer's disease group having generalized FER alterations. This effect was not seen in BVFTD in Parkinson's disease patients. Increased HEP modulations during post-interoceptive negative FER was present in healthy controls in Alzheimer's disease, but not Parkinson's disease in BVFTD. Anatomical signatures in all groups evidenced the involvement of the canonical cortical hubs, the insula, and the ACC. It also evidenced the involvement of the salience network and the integration of interoceptive emotional processes and the role of the EN and exteroceptive emotional processes, and the impact each have at the systems level. Disrupted connectivity associations were found between post-interoceptive negative FER and SN in BVFTD, and between post-exteroceptive negative FER and EN in Parkinson's disease, but these were both preserved in Alzheimer's disease, meaning that it does not rely on this network and it results in less specific deficits. This also suggests that multimodal signatures of interoceptive priming are compromised in BVFTD, suggesting interoceptive networks do not modulate the emotional processing in this disease. Altogether, behavioral results show the three distinct patterns of patient deficit, with only BVFTD showing a specific deficit of interoceptive priming on FER. To conclude, research on the interactions between internal body signals and emotion has a long tradition in cognitive neuroscience, neurology, and the related disciplines. Since emotional processing is partly modulated by the perception on visceral information, understanding how these multimodal signals integrate could prove for patients successful allostasis, that is, with efficient preparation of physiological need and internal states relative to the context. Our multimodal, cross-pathologic approach reveals new links between interoception and emotional recognition, pointing towards its distinct disruptions in BVFTD, the unspecific deficits in Parkinson's disease, and the preserved interoceptive priming effects despite generalized FER deficits in Alzheimer's disease. These results could possibly support theoretical accounts of functional synergies between the two domains while also opening a fresh agenda for basic and clinical innovations at the intersection of cognitive neuroscience and behavioral neurology. From a clinical perspective, with the fresh agenda open, new insights are offered about interventions related to body awareness and metacognition of emotion, interoceptive priors, and allostatic predictive coding addressing patients' emotional self-regulation. Our study showed important limitations which calls for further research. To begin, our design was based on a modest sample size. This was counteracted by the strict control of clinical variables as well as detailed diagnostic procedures and systematic assessments. Future studies should contemplate larger sample sizes and alternative designs allowing for exploration of important effects and patient group interactions outside the scope of our study. Because of the neutral, negative, and positive primes, the different negative emotions were not negatively assessed. Are you fucking